Welcome everyone. This is the second component of the module on study design and quantitative thinking. So, in this second component, we will be covering quantitative thinking, and there are two sessions in this component. So, let us start with the first session. Let us first revisit the scientific process in asking and answering questions in ecology. So, as we talked about in the study design session, based on our motivation, we first frame a question. Depending on the kind of question, we may also frame one or more hypotheses and predictions from these hypotheses. Once this is done, then we move on to study design, and we design our study using principles of effective study design. We then go out and collect data. After that, a crucial aspect is to analyze and interpret data and come to conclusions that can help us answer our question. So, in this session and the next one, we will be talking about good practice in analyzing and interpreting data. So, here is an outline of the two sessions on quantitative thinking. We will first talk about why we use statistical methods, and next we will talk about what are key statistical quantities that we commonly use. After that, we will talk about how we go about deciding what kind of a statistical model or method we might need given our study and data situation. And finally, what are some common mistakes that can lead to wrong results. So, I want to emphasize here that the goal of this component is to introduce the importance of quantitative thinking and to introduce key concepts. We will not be comprehensively covering or listing all possible statistical methods and how to carry out these methods. Okay. So, let us go on to the first objective, which is why do we use statistical methods. Just like in the study design component, here too we use statistical methods because we want to make generalizable and reliable inferences. By that, what we mean is that we want to be able to confidently generalize from sample to population, and what we uncover during data analysis and report, we want to be able to uncover signal that is characteristic of the population and not report a pattern that is noise, that is only restricted to this sample, and the next sample we collect will give us a different answer. So, to reiterate some terms that we used in the study design component, what we want to do is to get accurate, meaning we get to the population parameter as close to it as possible, and precise estimates, that is reliable estimates. If we were to do the study again, we would get very similar estimates. We would be able to locate that population parameter well. So, we want accurate and precise estimates of the quantities we are interested in. Before we go on to these quantities, let us talk about this accuracy and precision in greater detail. So, suppose the question that I am interested in is to look at how butterflies in a certain forest, how does their habitat use vary depending on how far or near a road the habitat is. That is, suppose my question is, how does road affect habitat use by butterfly? And I want to check whether butterflies prefer to use habitats far away from the road rather than near the road. So, I measure habitat use far from the road and near to the road using good principles of study design, and then I estimate the difference. So, this here is the difference. So, I estimate the difference in habitat use between far and near. And let us say we know that the true population mean is around 35, that is the difference in habitat use is about 35%. What I have shown you here is basically our estimated habitat use and the uncertainty, a measure of uncertainty around our estimate, an error bar here. So, this kind of a result is perfect. So, we have been able to get close to the true population mean and we have been able to estimate very precisely. So, our error bar is small. Another situation would be where our mean is accurate, it is close to the true population mean. However, the error bar, our measure of uncertainty in our estimate is very large, suggesting that it is imprecise. We have not been able to locate that population mean very well. Here are two other situations. First, a case where our estimate here, our estimate is inaccurate because it is very far away from the true population mean. It is also imprecise because the error bar is very large. And finally, we might get again an inaccurate but very precise estimate. So, our error bar, our measure of uncertainty is very small. However, notice that the error bar and the estimate are very far from the true population mean. So, the latter two cases are particularly dangerous because they give us an overestimate of the true effect. Okay, let us consider a different situation. So, that was a situation where the true population mean was 35%, that is butterfly habitat use, the difference between far and near is 35%. So, they have a very strong preference for habitats far away from the road. Now, let us take up an example where the true population mean is 0, that is there is no real effect of distance from the road on butterfly habitat use. Butterfly habitat use is similar both far away from the road and near the road. Now, if we get an estimate, so we go ahead, carry out our study, calculate the difference between far and near in butterfly habitat use, and we get an estimate that is very close to 0. So, we have got a accurate estimate and it is also very precise because the error around the estimate, the estimated error around our difference in habitat use is very small, our uncertainty is low. So, this would be an ideal result. However, we could get these other situations here again. So, an accurate but imprecise, large error bar, inaccurate because the mean estimate is far away from the true population mean, inaccurate and imprecise, inaccurate but precise. Again, just like in the previous case, these last two situations are particularly dangerous because they give us a very biased view. So, here for example, we would be wrongly concluding that butterflies prefer habitats far away from the road. Basically here noise in the sample would be reported as signal, as something characteristic of the population. Now, in both these cases, in both these examples, I have told you that we know the true population mean. However, typically we do not, right? Typically in a question like this, where we are interested in understanding how roads impact butterfly habitat use, typically in such cases, we don't have an idea of what is the true situation in the population, what does the true population mean. So, how then do we know whether we have accurate and precise measures? There are certain practices in study design which we talked about, which can increase accuracy by reducing bias and increase precision by letting us reduce noise. Similarly, in statistics also there are good practices that can help increase accuracy and increase precision. That is what we will talk about in the last part of these two sessions. Okay. Now, when asking and answering questions in ecology, there are some common tasks that we need to do. So, one common task is measuring a single quantity, maybe in a single place or in a single point in time. For example, how many birds are there in this grassland? Another common task is to ask what is the difference in species richness, that is the number of bird species, between large and small habitat patches. So, this is a task that involves comparing a quantity between two situations or more. A third common task is to estimate relationships, that is, for example, how is species richness related to the area of the habitat patch. So, here we might either be interested in describing patterns in the relationship, so patterns of covariance or correlation among these conditions or traits, or we might actually be interested in explanation or causal inference. That is, we might be interested in seeing how species richness is affected by the area of a habitat patch. So, for these three types of common tasks, which is measuring a quantity like how many bird species are there in this grassland, or comparing, for example, what is the difference in species richness between areas that are small and areas that are large, or correlating how is species richness related to the area of the habitat patch. For all of these, there are some common statistical quantities that we will use to achieve these tasks. First, we want unbiased estimates, that is, from our sample we want to calculate estimates that are a good reflection of the population. Just like in study design, where there are certain principles, if you follow you can get unbiased estimates. Similarly, in statistical methods also, we can figure out what can give us unbiased estimates. So, the mean, for example, is a good unbiased measure of getting an unbiased estimate. In some cases, the median or the mode may be a better unbiased estimate. This is for the first question about how many bird species there are. Similarly, we might calculate the mean difference or calculate the correlation or some other estimate of the relationship. Now, the second statistical quantity that we are likely to be interested in applies to cases where we are either comparing or estimating relationships. So, we might be interested in what is the difference in species richness or how is species richness related to the area of the habitat patch. And here we are not just interested in saying there is a difference or yes there is a relationship. To make conclusions about biological importance, about ecological importance, we are usually interested in the size of the effect. What does that mean? We are usually interested in the magnitude of the effect. For example, let us take up this question of what is the difference in species richness between large and small habitat patches. Here, This is a situation on your left of a case where there is a large difference in species richness between large habitat patches and small habitat patches. So, a difference on average of 10 species between large and small habitat patches. On the other hand, look at the situation on the right where there is only a very small difference in habitat species richness between large and small habitats. Hardly 3 or 4 species more on average in large habitat patches compared to small habitat patches. So, this is a reflection of how important ecologically is the area of a habitat patch. What is the area effect? Let us take this example again of how species richness is now related to the area of the habitat patch on a more continuous scale. That is, on the x axis now, take the image on the left, on your left. On the x axis we have habitat patch measured in square kilometers, and on the y axis we have the number of species, that is species richness. So, the plot on the left shows you a case where species richness is quite highly correlated with habitat patch size. This would tell you that habitat patch size actually explains a lot of variation in species richness. However, the case on your right shows you a case where as habitat patch increases, the correlation with number of species is quite low. This can be estimated through coefficients like the Pearson's correlation coefficient, for example, which shown here is high, correlation of 0.9, shown here low, a correlation of 0.5. So, here the effect size is the strength of the correlation between number of species, species richness, and the habitat patch size. However, for a question like this, just the strength of the correlation may not be sufficient. Instead, what might be important for us ecologically to answer our question might be the magnitude of the slope. That is, by how much does species richness change, that is the number of species. How much does species richness change as habitat patch size increases? So, on the left you have a case where for every 10 square kilometer increase you get an additional 9 species. So, that is a large effect. On the other hand, on the right you have a case where for every 10 square kilometer increase, the increase in species richness is comparatively small. It is only 3 species, which means in the whole study area from the smallest, if you look at the smallest patch size to the largest patch size, you only have an increase about 5 to 8 extra species as you go from the smallest to the largest. On the other hand, look at the case on the left where as you go from the smallest to the largest, you have a huge increase in species richness from around 5 species all the way to around 40 species. So, as an ecologist, if you are interested in this area effect on species richness, just declaring that area affects species richness is not interesting. What would be really interesting is to know by how much is area affecting species richness. So, the magnitude of the slope in this case. The next quantity that we are interested in is the confidence in the estimate. So, whether our question is about how many bird species are there in this grassland, or what is the difference in species richness between small and large habitat patches, or how is species richness related to the area of the habitat patch, in all these cases we get a mean estimate, mean species richness or mean difference or mean correlation. However, we are not interested in the sample mean for itself. We are interested in the sample in what it can tell us about the population. So, we are interested in the population parameter. So, we usually use the term parameter to indicate the true population value, and we use the term estimate to indicate the calculated value from the sample. So, here the parameter, population parameter would be the species richness or difference in species richness or slope of species richness with patch area and so on. So, what we would like to know is when we calculate our sample estimate, say the estimated difference in species richness, how close is it? How good a guess is the sample estimate of the population parameter, the true value which is not known, which we are trying to get at. For this, what we want to know is how precise is the estimate, what is the precision of the estimate. If the estimate is very precise, then we have been able to locate that true population value, that population parameter well. If the precision is not very high, then there is a lot more uncertainty. We are not so confident about where the population parameter lies. To measure precision, one of the most common quantities is the confidence interval. So, when we calculate the confidence interval and we get this interval that this is the sample estimate of the difference in the number of species between large and small habitat patches. So, the mean difference is 10, and if we then calculate the confidence interval and find that it ranges from 8 to 12, then we can say that we have a fairly precise estimate. The true population parameter lies somewhere between 8 and 12. So, there is less uncertainty, more confidence. On the other hand, if we get a wider confidence interval ranging from 4 to 16, then here we have less precision, more uncertainty. The true population parameter lies within a much larger range of values. Since the confidence interval is really important, probably one of the most important concepts in statistics, let us spend a little more time understanding confidence intervals. Now, typically when we carry out our study, so here I have shown you an example of carrying out the study on comparing species richness between large and small habitat patches. So, I have sampled, say for example 15 large patches and estimated species richness in each of those 15 patches, and then I have focused on small habitat patches, and here are the data from 15 small patches. The difference in the mean species richness between these two samples, the large and small habitat patches, I find that difference to be 7. It is important to remember that this is one sample, one possible sample from the larger population. If I were to carry out this study again, I am going to get a different data set, a different set of values for large patches and a different set of values for small patches. And now the mean difference in my sample is 8. So, it is important to remember that each time we carry out our study, we are getting one guess at that true population parameter, the true value that we are trying to get at, right. So, if we were to do this study many times, each time we calculate a sample mean difference, and if we were to then plot that out here. So, what this shows you is a frequency distribution of mean difference between large and small habitat patches. So, these are mean difference, the sample estimates. So, the distribution of these means is called the sampling distribution of the parameter, and this gives us a lot of information. This tells us if we were to do the study again and again, will the different estimates of that population difference, will we get similar values or very different values? If we get similar values, then we know we are getting a good guess at that true population parameter. If we now get very different values, then we know that we have not been able to locate that true population parameter very well. So, the sampling distribution of the parameter is affected by how much variation there is in the population. If there is a lot of variation, then pinning it down gets difficult. It also depends on our study design. Very important aspect of study design that it depends on is sample size. So, if we increase sample size, the panel on the right shows you the sampling distribution of the same parameter, which is the difference in species richness between large and small habitat patches. It shows you the sampling difference when you have a large sample size. So, you can see you have a narrow sampling distribution, which means each time you do the study, you will keep getting very similar guesses of the true population parameter. Your sample means will be very similar to each other. So, a narrow distribution means you have high precision, high confidence in your estimate of the population parameter, low uncertainty. On the other hand, a wide distribution tells you that you have low precision, low confidence and high uncertainty. To capture precision, the measure that is typically used is the standard error. So, what is the standard error? It is the standard deviation of the sampling distribution. So, remember from basic statistics that standard deviation of your sample tells you how much spread there is in your data set. The standard deviation of the distribution of means, this is the sampling distribution, here is the distribution of your sample means. The standard deviation of the sampling distribution is called the standard error. Wide standard error would mean imprecise and narrow standard error would mean a very precise estimate of the statistic you are interested in. Here for example, mean difference in species richness. Now, we can use the same concept to calculate the confidence interval. So, given the level of confidence you are interested in, you can then calculate the range of values, the interval that captures the central specified distribution of your sample means. So, for example, if you are interested in the 95% confidence interval, what is the interval? What is the range of values that captures the central 95% of your distribution? So, in this case, the range 4, a difference of 4 species to a difference of 12 species between large and small habitat patches. 4 to 12 is the 95% confidence interval of your estimate, which was the mean estimate was 10. So, just to repeat, just to recap here, the 95% confidence interval is the interval that covers the central 95% of the area under the curve or the central 95% of your sampling distribution. You can calculate confidence interval with different levels of confidence. So, if you want a 90% confidence interval, then what you are trying to do is to find the interval which captures the central 90% of the area under your distribution, that is the central 90% of the sampling distribution, the values of the mean difference between large and small habitat patches. Now, the greater the confidence, the wider the interval, because you want to capture more and more of the probability of the sampling distribution. Okay. So, usually you would not see the sampling distribution, usually the way your estimate and confidence interval are shown is in the form of an error bar. So, for example, same example, we have a mean of 9 and a 95% confidence interval of 5 to 11. So, this would usually be shown as the difference in species richness between large and small. 0 would mean no difference, negative value would mean more species in small habitat patches, positive value is more species in large habitat patches. So, here you can see we have got a mean of 9 and a confidence interval ranging from 5 to 11. So, you can think of the confidence interval, the intuition here is that it is the range, plausible range within which that unknown population parameter that we are interested in, it is the range of values within which that unknown population parameter is likely to fall for a given level of confidence, here 95%. However, it is useful to know specifically how it is calculated and technically what it means. So, technically what it means, if we are to take the 95% confidence interval for example, if we were to repeat the study many times and each time calculate the 95% confidence interval, the true population parameter will occur within these intervals 95% of the time. Let us visualize this. If we are to carry out the study many times, and I have shown you that. So, the first time you carry out the study, you calculate the sample mean, here it is the mean difference and the confidence interval. Second time you have a different data set, it is from the same population, but just because of sampling you get a different data set, different mean difference, different confidence interval and so on. So, you carry out the study many times, each time you calculate the sample mean and the confidence interval around it. Suppose we know that the true population difference is 9 species between large and small habitat patches. The 95% confidence interval represents the interval in which the true population parameter will occur 95% of the time. So, you can see in this example all these intervals, the true population parameter of 9 falls within all these intervals except for one in this case. So, in this iteration of the study, the confidence interval does not include the true population parameter. So, technically the confidence interval is the percentage, represents the percentage of all intervals that contains the population parameter, or another way to think about it, it is the probability that the interval produced includes the population parameter. So, 95% confidence interval, there is a 95% probability that the interval you have produced includes the population parameter. Remember, in any one iteration, the interval either holds the population like in these cases or not. Here the population parameter does not fall in it. So, in any one iteration, the population parameter is either located in that interval as in these cases or not as in this case. So, let us get back to the intuition. So, the intuition is the confidence interval is the range within which that unknown parameter is likely to fall. A wider confidence interval indicates very high uncertainty, low confidence. So, the true population parameter can fall within a very wide range of values from no difference all the way to a very large difference between large and small habitat patches. A narrow confidence interval indicates that the true population parameter is you have been able to locate it quite nicely, quite precisely. It is likely to fall within a narrow range of values. So, how do we actually calculate the confidence interval? What I have talked to you about till now is more a thought experiment. We do not actually do our study many times. We typically do our study only once. But from that one carry out of our study, we can calculate the confidence interval. So, one way to do it is to consider our sample as the population and resample from our data set. Each time we sample our data set, calculate the statistic, for example, the mean difference between large and small samples, habitat patches, and create a distribution of the statistic. Then for say a 95% confidence interval, we find the interval within which the central 95% of the distribution of the resampled statistic lies. So, this is called the lower confidence limit and this is called the upper confidence limit. A more common way of calculating the confidence interval, which you are probably familiar with, is if we can make some assumption about the sampling distribution of our statistic. What is the sampling distribution? Remember, the sampling distribution represents the distribution of sample means, the distribution of our estimates if we were to carry out the study many times. Now, if we can assume that the statistic that we are calculating, we are estimating, if the sampling distribution of that can be assumed to be normal, then we can calculate the standard error using concepts from the normal distribution. So, now our measure of precision, the standard error, is calculated as the square root of the sample variance divided by the sample size. So, another way you would have seen this is as the standard deviation divided by the square root of sample size. The good thing is that for many ecological cases, the estimate, the statistic that we are measuring is very often we can assume it to be normally distributed. This is because of the central limit theorem. I will let you go and look that up. But because of the central limit theorem, we can actually assume the sampling distribution of the statistic to be normal quite often in our ecological studies. We then use this calculation, this way of calculating the standard error to construct our confidence interval. So, again, just as we discussed before, let us reiterate that our measure of precision, the standard error, depends both on variance, which measures the variation in the population, and sample size, which is the study design, which is a feature of the study design. So, as the variation in the population increases, variance increases and our standard error will increase. If our sample size is small, then we do not cover that variation well and so our precision is low. If our sample size is large, then we cover the variation in the population well and we are able to estimate the quantity of interest, we are able to estimate it very precisely. So, here I have shown you two cases, one case where we have very narrow distribution with a small standard error when sample size is high, and for the same population if your sample size is low you get a wider distribution with a wider standard error. We then calculate the confidence interval as our estimate, say the mean difference between large and small habitat patches in species richness. So, the estimate plus or minus the critical value times the standard error. What is this critical value? If we can assume that the sampling distribution of our statistic is normal, then we can find the number of standard errors on either side of the estimate which covers the specified probability of including the parameter. So, so we know, for example, in the standard normal distribution, which is what is shown here, we know for a standard normal distribution where you have mean of 0 and a standard deviation of 1, we know that approximately 1.96 standard deviations on either side of the mean covers 95% of the area under the normal probability curve. So, we use this property to say that if our sampling distribution of the statistic, we can assume it to be normal, then the mean, the estimate plus or minus 1.96 times standard errors will now cover 95% of the probability. So, this interval will now represent the 95% probability of including the population parameter. So, the confidence interval is crucial to interpret the estimate. So, going back to our question, what is the difference in species richness between large and small habitat patches? You can see that the mean difference here is 9. However, confidence intervals will change our conclusion. Let us take 3 examples here. So, we have 3 examples. In all these examples, the mean is the same. The mean difference in species richness is the same. However, take these 3 examples. The first example, the 95% confidence interval indicates a very small range of values in which the true population parameter is likely to lie. So, here we can safely conclude that there is a substantial difference in species richness between large and small. We have located that population parameter well. Take the next case. Here you have a much wider confidence interval which includes both no difference all the way to a very large difference. So, here we have very high uncertainty, very low precision. We have some evidence that there might be a difference in species richness, but we can't be confident about it. The third case, same mean species richness difference, however, an even wider confidence interval which includes both cases where small patches have more species than large patches, all the way to where we have large patches having more species than small patches. Here we cannot reliably make any conclusion about whether truly there is a difference in our system, in our study system, in species richness between large and small patches. Let us take another example. We had the case of how species richness relates to the area of the habitat patch. Here I have shown you that same estimate with a confidence interval around the estimate. Here we have managed to locate the estimate, we have managed to calculate the estimate with precision, because the confidence interval here around the slope represents the effect between number of species and habitat patch size. The confidence interval around the slope is narrow, indicating that yes, you could have slightly shallower and slightly steeper cases, but that true population parameter, the true slope, seems to be captured quite well. On the other hand, take this case where we have a very wide confidence band around our regression line. So, here apart from our mean estimate of the slope, the confidence band indicates that the true population parameter could be much shallower, close to 0, or much steeper, indicating that we have not managed to locate that true population parameter very well. The range of values that it could plausibly take ranges from negligible slope to very high values of the slope. So, to summarize this first part of the quantitative thinking component, we have gone through why we use statistical methods, we have talked about what are some key statistical quantities that we commonly used, and here we covered getting a reliable estimate, we talked about effect size, and we talked about a measure of confidence in our estimate. So, this ends the first part of the component quantitative thinking in the module study design and quantitative thinking. Thank you.